Hello there everyone. In today's lab, we will be doing the antibiotic sensitivity test, which is a test to measure the ability of antibiotic or other antimicrobial agent to inhibit or kill the growth of microorganism. In this test, there are two terms, the minimum inhibitory concentration and minimum bactericidal concentration. The minimum inhibitory concentration, which refers to the smallest amount of agent to inhibit the growth of microorganism, and bactericidal concentration, which need a small amount of agent to kill the microorganism. There are methods that are used, the disk diffusion method and quantitative E-test and dilution. The method that we are going to apply today is disk diffusion method by using a special agar called Muller-Hinton agar. In this agar, types of antibiotics are going to be placed with a inoculated tested microorganism, and then we are going to incubate the plate at 37 Celsius for 16 to 18 hours. If an organism is sensitive to an antibiotic, a clear zone will be produced around the disc, and then we are going to interpret the results. Okay, first we are going to make a bacterial suspension, which take 2 to 3 colony of bacteria. And mix it with normal saline. And then we are going to compare it with the McFarland standard to make sure the turbidity is 0.5. After that, we're going to take a swab and take the suspension, bacterial suspension. And spread that evenly on the Muller-Hinton agar, which is a special agar for this test. The bacteria should be spread evenly. And then we are going to place different antibiotic on the Muller-Hinton agar to determine which antibiotic will inhibit or kill the bacteria. The inhibited area will produce a clear zone around the disc. Now we are going to place the antibiotic. We need to place it somewhere that when the zone is going to be produced, it will be visible away from the and then we are going to let it dry for 3 to 5 minutes. After that, we are going to incubate it at 37 Celsius for 16 to 18 hours. And then we are going to interpret the result. As we said before, if the antibiotic has the ability to inhibit the growth of microorganism, it will produce a zone around the disc, and then with a roller we're going to measure the inhibition zone.